Good afternoon. My name is Christina Drake. I'm the founder and CEO of Kismet Technologies, and I'm going to be talking about accelerating technology development for breakthrough innovations using a project that I am working on with UCF to commercialize a product as an example for how to practically do this accelerated development. And so if you've been involved in innovation and product development, you're probably familiar with this graph, which is the innovation life cycle of products. And on the far left side, where you have discovery, which is where something is discovered that potentially has benefit in the real world. That's typically done at research institutions such as universities because of their academic mission to add to the knowledge base of mankind. And then on the far right, you have companies and large businesses that have a lot of capital and infrastructure and ability to monetize products. And in between those two, you typically have what's known as the valley of death, which is that opportunity space between discovery of some phenomena and the ability to turn that into a product that can meet some need in the real world. And typically in that space, stereotypically, you would have a startup or entrepreneur in that space. And so they tend to be resource non-intensive. And so this picture down here of sort of the stereotype of an entrepreneur in a garage, sort of like Steve Jobs, is typically what comes to mind. And so it's tempting to say that this is just resource due to money is the reason that you see this valley of death. But if you look at research funding in the United States, and you look at basic research versus applied research, you actually see that in the United States, and so this is from 2018, a report that was given to Congress, that actually applied research was funded more than basic research in the United States. And if you think about what universities, their purpose is with basic research, not all of that basic research is done with the intent of innovation. Some of it is just exploratory in nature. And so not all of that is done within the innovation cycle. So you can think of that as that being an overstatement of the money given to basic research for innovation. So funding is probably not the main driver in terms of lack of resources for getting across the valley of death for something that is a potential innovation. And so Forbes has a stat that 9 out of 10 startups will fail, and that this is primarily lack of market product fit. And market product fit can be things like having a solution, looking for a problem in the market and not ever finding what that problem is that it can solve, or not making it to the market at the appropriate time, maybe making it there too late. And so another way to look at the innovation life cycle is to use technology readiness level, which is what NASA uses to describe the innovation life cycle. And it has 4 different phases. So you have basic research, applied research, tech development, and prototype and system development. And there's 9 technology readiness levels to get something to full commercialization. And if you're talking about physical or hard sciences where something is being made, so not software, and especially if you're talking about in life sciences, this life cycle is typically between 8 and 15 years. So from the time that something is realized as being innovative or discovery in basic research, it can take a long time, about a decade, to get it into the market. And so a lot of this has to do with the very serial nature of how we get those new breakthroughs passed along through that initial life cycle of development. And so this valley of death now overlaid with the different institutions that typically are involved in these different phases of development life cycle. This valley of death occurs right at about the point where you have a prototype. So typically in serial approaches, university researchers, they know the science, they understand it, and they have institutional resources like equipment, students, but they may not necessarily understand the market need. So what they focus their research on might not fully answer the questions for whether or not something would have meaning in the market. An entrepreneur may have an idea for solving a needed market problem, but it needs to understand the science better in order to really do the tech development and do it well. And so instead of using a serial approach, you can use a parallel development approach where you have 3 iterative design cycles between the 4 phases in order to help compress the development and focus it better. And so when you're able to do that, and I'll use our company product commercialization process as an example of how to do this. You can compress the early timeline so that you at least get rid of time factor to market as one of the potential failure mechanisms. But then I think when you also do that and you're able to leverage the infrastructure of universities to help with some of that initial exploration to focus the tech development, it lessens the severity of the valley of death, and you're able to make good use of both resources from a university and from a startup to make progress really, really fast. And so backing up a little bit to kind of talk about who the team is. So my company, Kismet Technologies, we create advanced materials to help people live healthier and safer lives. We're part of the UCF Business Incubation Program, and we are also part of the Mass Challenge Life Science Accelerator. We are funded by the National Science Foundation and supported through the Florida High Tech Corridor through grant matching with UCF, and then also as part of their life science cluster, and we're also a part of BioFlorida. And the company is supported through private angel investment for commercialization and regulatory approvals. And so our team that we work with at UCF, Dr. Sudipta Seal, who's a world-renowned expert in nanoceria for bio applications, Dr. Griffith Parks, who is a world-renowned virologist in the School of Medicine, and then also Dr. Melanie Kothup, who works in implant infection and helps us with bacterial studies. And so last year, at the beginning of the pandemic in April, the team actually did not originally include Dr. Kothup. It was myself, Dr. Seal, and Dr. Parks. We submitted an application to the National Science Foundation for creating a rapid-acting disinfectant to stop the spread of COVID-19. So in April of last year, it was not really clear whether or not surfaces were a vector of transmission for COVID-19. And so we had focused, that seemed like the near-term product or the near-term need in terms of viral disinfection. And so that's what we had focused our project on, was can we create a fast-acting spray that worked in under 30 seconds to get rid of viruses. And because it was an NSF project, we had to have strong scientific rationale and a hypothesis to go behind that. And so we were awarded in August of last year, and by September of last year, we had began material synthesis. And so here's a TEM of some of the material that was created. And very soon after, we began viral testing in Dr. Park's lab. And we focused on liquid test because we were focused on a fast-acting disinfectant that could work on surfaces. And we got really good results. So we were able to see that you could reintroduce viruses to a liquid that had this nanoceria in it, and that it would actually disinfect it. And so on the top here, you see coronavirus, and on the bottom here, you see rhinovirus, which causes the common cold. And so we actually, with these tests, saw that we had very good efficacy with the materials that had initially been created. Simultaneously, while we were doing the materials characterization and the viral efficacy test, we began doing customer interviews on performance requirements to understand more in the market. How would a customer, like what would they want, someone in healthcare? What would they be looking for in a disinfectant? And actually what we learned overwhelmingly was that customers did not care about how long it took for something to disinfect. That they really cared about was whether or not it could be residually disinfecting and take basically the burden of time to have to constantly go back and disinfect. If we could remove that, that is something that would really solve a problem for people in healthcare in the world. So we began looking at EPA requirements, regulatory requirements, and so this is all happening while we're doing our initial synthesis and initial viral efficacy tests. We're doing these all in tandem. We began looking at the EPA requirements just to register as a residual disinfectant. And you have to have a performance standard against bacteria for your material or your residual disinfectant. And the original plan had been, well, we'll outsource this to a third-party lab and we'll get them to do the test. So we thought that was I had thought that that would be a really easy, quick way to get results back. After working with a few labs and getting some quotes back, we actually found out that for one test, it was 4 liters of material for one bacteria for one test. And so I have a picture over here, and this is from Dr. Seal's work. We make very small quantities of material right now because we had just started synthesizing it. So 4 liters was actually not feasible. We consume all the material we make, we test all the material we make. And so we made 2 pivots in January of this year with our project. The first was we began focusing more strongly on dry film challenge of the material. And in doing this, we were able to see that actually at a lower concentration than what we had been doing in liquid, we saw even better results than what we had initially saw with the liquid. And so in this one, this is from Dr. Parks. You see that we've challenged a film or a slide that has dried nanoceria on it 4 times with virus without removing it. The first 3 times you see eradication, and the 4th time you see full disinfection. So even more spectacular and fantastic than what we had found the first time. And then we also saw that with coronavirus, if you see down here, I don't know if you guys can see my cursor, you see that there's an AGCNP1 and an AGCNP3. We had actually started with 2 forms of the nanoceria, and originally in liquid form, the AGCNP1 performed well against coronavirus, and this AGCNP3 performed kind of okay. But in dried form, this AGCNP3 was spectacular. It eradicated the coronavirus, no infectious virus left. And so that was a pivot that we made that actually showed where the true, in terms of quality of the material, came about. And it was from actually doing customer interviews. It pivoted how we were doing our research to address what was needed in the market. And we found that we actually had maybe focused not in the direction where our material probably performed best. And then the other pivot that we made was, instead of giving out a lot of material to a third party to do our bacteria test, we brought Dr. Melanie Kothup on board to help us with our bacterial test. And what was great about that was not only did we get to learn whether or not our material had efficacy against bacteria, we now could understand more the science behind why and what was going on, and make tweaks to our experiments to understand better the behavior of the material. And so on the left here, you see Pseudomonas, and on the right, you see staph. These are both required by the EPA in order to register as a disinfectant, even if you're residual. And you see we re-challenge multiple times. We have great efficacy for both Pseudomonas and staph. The other fun thing down here, and this was from between Dr. Kothup and Dr. Seal's group, but they were able to put Pseudomonas on dried nanorad and actually see that by the 15-minute mark, the Pseudomonas has actually begun to lose its integrity. So we have an indication that this disinfection behavior is happening much earlier than maybe what we're seeing from the 2-hour test that we were doing. And so, now we've turned our focus away from just coronavirus to something much bigger, to hospital-acquired infections. So in the US, 1 in 30 hospital patients will get a hospital-acquired infection. And US hospitals lose $30 billion a year to HAIs. When we had started the project, we had focused on just surface disinfectants, which is a globally a $4 billion a year market. But by listening to customers and kind of refocusing what we were doing in the research and science, we found a problem that did not have a good solution to it, and we're able to see fairly quickly whether or not we could address that problem. And also because we had done these customer interviews and understood that really what mattered to customers was whether or not something could stay on the surface for a long time, we redid the formulation that would hold the nanoceria down on a surface to last as long as possible. And in our first prototype, we have a formulation that lasts past 7 days in wet wear abrasion test. So it's mechanically sturdy enough to stay on a surface for that long. And now we're working on other variants of that, working on things like optical quality of the actual film, the way that the film feels, so things that customers might care about when people encounter this residual disinfectant on the surface. And so this is a little bit of a complicated chart, but this shows 3 of the first 4 phases of product development. So basic research, applied research, and product development. And what I'm showing here is basically the feedback, the way that information flowed and informed other portions of our development and where the feedback loops were. So for example, material synthesis and characterization, those were fed into virucidal efficacy and bacterial efficacy test. It also fed into the formulation development because it had to be something that was soluble and could work with that material. The formulation development and the properties of the formula fed into our market research. Information that we learned from that flowed back and helped us to basically focus our research and our formulation to meet market need, and so on and so forth. And now in spring of this year, we had a fully working prototype for the residual disinfectant, and we're now working on optimization. And so being able to co-locate the basic research with the applied research with some of the product development led us to product pivots to market need early on, so that we weren't spending time in areas that maybe didn't have, much of a market pull for what we were working on. So we started out as a fast-acting surface disinfectant focused on coronavirus deactivation. And we had pivoted to a long-lasting residual surface disinfectant focused on hospital-acquired infections. And so basically in a 10-month period, we were able to go from a TRL1 to a TRL4. And so for anyone that has worked in materials development or life sciences, that is extremely fast. I think for what we did, that would normally have been a 3-year to 4-year time period, and we were able to do it fairly quickly. We've also had very good traction with potential customers and people who have basically let us know that they are ready to license once we are able to get EPA registration. And so my company is currently right now going through the fundraising process to get us through that regulatory hurdle, but we have good market fit with what we're doing. But the broader impact of what we're working on, we have the ability to drastically reduce hospital-acquired infections, which is really important. Another area is potentially reducing antimicrobial resistance, or AMR. It's a really big public health challenge in the US, but also globally. And we've also, related to AMR, our team was invited to submit 2 proposals to the International Space Station. And one of those looks at antimicrobial resistance with bacteria with our material. Some of our other base research findings may have applications in other areas. One of the things that we learned, and this was because we had to have certain tests for the EPA, was that the material that we had synthesized appears to be not only friendly to tissues, but may help their viability. So if you have something that could help tissues and also kill bacteria, then in wound healing, you have potential application there. And so we have a lot of other areas that we're now looking in based on our research findings. So in terms of accelerating innovation, running parallel innovation phase tracks can help you to fail fast and pivot quick early on, so that you're not spending time researching and building things that basically don't have market fit. It's a more efficient design cycle. So we were able to, with $256K from NSF and $250K from an angel investor, basically get to our first prototype really quickly. And then we have a productive body of knowledge that we've acquired. So now for things like wound healing and other applications, we can explore those and with the understanding of the science for what we've worked on. Keep incentives in place. So joint funding is important. I think that helps you to feel like you're on a team with the people that you're developing with. Publish on the basic science. So don't be scared that if you publish, somehow people will steal your IP. I think that's a I think that's a trap to think that, and keep exploratory science with the targeted science. So you should have goals, but you should also make room for fun in the exploratory science. And make best use of each group's core function so that it'll keep frustration down and the research exciting. And I think that in our group, we do have a lot of fun and we have very animated conversations. So overlapping that innovation development cycle and design iteration will compress the timeline. And I think if you're looking to bring something to market out of basic research, can help that success rate through that valley of death. And I will take questions.